Hello, everyone. Let's start Cells and Molecules Part 2. So let me do a screen share on that right now for you. All right. So the last time we were talking about DNA, I mentioned the discovery of DNA by Rosalind Franklin. We're talking a little about its basic structure, how it was elucidated. Now we're really going to be talking about it is how it's put together. Really how these products of DNA, which are the amino acids, right? How those are put together and how these function in their body and how those things actually change too, and how we can decipher that. What sort of tools we have to sort of decipher the DNA code. Okay. So what we know from Rosalind Franklin is he discovered that there are 4 base pairs, right? Nitrogenous base pairs, A, C's, T's, and G's. But remember, these are not amino acids, right? These are not the products, right? They're just part of the code. They exist on each of the strands of DNA. So let's go back and have a look at DNA. You can see that A, T, C's, and G's, and there are 2 strands, right? You can see A, T's, and G's a complement assortment of one strand, and A, T's, and G's a complement assortment of the other strand. Now, the genetic code itself only exists on one strand. For our purpose here, strand 1. The other complementary strand is really where they come together as a protective strand, right? The other strand is just to provide some structure and function to it. And it can also be unzipped to actually expose the real genetic code. And it can be unzipped to expose, like, say, strand number 1 for the purpose of copying the DNA, for the purpose of creating different cells, such as cells for my body, which are my somatic cells, or sex cells, which go into reproduction. And it's in the copy we're going to see that changes are made, which are known as mutations, which are the cause of evolution for things to actually change here. Okay, so now we sort of got the basic patterns here. Now, every 3 of those letters of one strand represent a unique and individual amino acid. And remember, we're talking about a very long linear piece with one strand of DNA. So there's 3 letters followed by 3 letters, followed by 3 letters, followed by 3 letters, all represent amino acid. And these amino acids, there are 20 different ones of these. There are 20 different amino acids. There are only 20. Okay, they're all 20 in different sort of order, which roughly represent all the proteins in your body. This, taking 20 different things, mix matching in different ways and making very long chains out of them. And of course, that's coded within the DNA itself, represented by 3 letters at a time. Okay? Well, when the body, right, takes the DNA, copies a strand of the DNA, and delivers that strand to the ribosome in a copied form, the ribosome will read the strand and begin to sequence the amino acids. So it'll see the 3 letters, like DNA says these 3 letters, and it will match the particular amino acid this one's designed for, 20 different ones. And then it'll look at the next 3 letters, okay? And then it will say, okay, I need to say this is tyrosine. These 3 letters represent tyrosine, and this next 3 letters represents arginine. So I'll take a tyrosine and arginine and bind them together. When I bind them together, this is exactly what a ribosome does. We create a dipeptide bond. And if you put a lot of dipeptide bonds together with a lot of amino acids, called a polypeptide is a technical term, and really we're trying to get long chains of polypeptides, fold them up into things which are usable for the body. And they can be used as structure, build the framework of the body, the raw materials, or they can be used as enzymes, things with molecular function and molecular tools. That's what we're really interested in. Those really do the bulwark of what we're talking about when we're building a body, right? I'm building organisms, right? So once we can sterilize by osmosis, we can sort of get the process that's happening here too. So we remember that only the codes on the inside, the outside, those P's and S's, the sugars and phosphates, is nothing but for structure, right? We see the genetic code just on one side, strand number 1. It's in a linear format, T, G, A, C, or whatever it is, every 3 letters at a time. Now, the complementary strand has some rules to it too. Notice how it pairs across the bonds, okay? So only T's pair with A's and G's pair with C's, okay? So notice that, or A pairs with T's or G pairs with C's, whichever one of you want to go back and forth. That's the pairing rules. Anytime I see a T, I'm going to see an A on the other side when I'm looking at DNA, a DNA molecule, okay? If I see a G, I'm going to see a C if I'm looking at a DNA molecule. And so that's sort of interesting how the pairing rules sort of work between one strand and another. And that's going to be important a little bit later on when we're looking at how a copy of this is made and then sent to the ribosome, okay? Now, but for now, let's see how geneticists and anthropologists, how do we look at this stuff and sort of talk about the changes in this system? How do we record them? How do we find them? And how do we make sense of what they mean? All right, so this is a code book. of all of the letters in your DNA that represent amino acids. This is it. This is all the combinations of your entire DNA. All 3 billion letters are just combinations of 3 letters like this. Okay. So for alanine, there are 20 different amino acids. There's alanine. There's just one of them, right? So look at alanine. You can get, and what we got to make sure we're only going to be looking in the light colored columns, the white columns, not the green columns. Okay. The white letter columns have the DNA. That's a DNA codon. That's 3 letters. Okay. Not the green stuff. That's mRNA. That's the copy of the DNA, which we'll talk about a little bit later. Okay. All right. So for alanine, we can get alanine by going CGA. Look, wow, if I see CGA, look at my DNA on strand number 1, there's alanine, right? Let's say it was followed by a CCA, like over the glycine. Oh, there's glycine. But look at how many different ways there is to get alanine. There's 4 different ways. How many different ways to get glycine? 4 different ways. How many ways to get to proline? 4 different ways. Why? Well, we have to go back to Rosalind Franklin again. Remember what she said? She said, listen, their genetic code, is comprised of 4 things, right? But because it can be paired in 3 ways, triplets, right? 4 things, that's 4 cubed. How many different things can we represent? 64. See, we've got enough amino acids, triplets, 3 letters, combinations to represent 64 amino acids. We only need 20, but we can do 64. That means we've got redundancy. That means, down here when we're looking, that means alanine can be expressed in many different ways, which is what we see, okay? So, and of course, there's varying degrees of this too. Like, arginine, there's 6 different ways, but for asparagine and aspartic acid, there's like 1 different way or 2 different ways to get, that amino acid, as far as representation, the code for it, right? So when reading along DNA, we're looking just for that. That's our code book, okay? But one important thing is that the body needs to know the machinery that copies this stuff from the DNA, takes it to the ribosome, needs to know where to start. Well, where does this protein start at? Which is really the gene. Okay, so a gene is a protein, a full protein coding stretch of DNA that expresses 1 complete protein. That's a gene. And you've got about 23,000 genes. Okay, so 23,000 tools and pieces of raw material it takes to put you together, which is about the same as a Tesla. Right? I'm a Model T. I don't know what model you guys are, but I'm a Model T. Right? So I remember that, about 23,000. Each one is a protein coding gene. We have to know where to start. Okay, so the body sees a particular letter sequence, and it's called an initiation codon. You can see it right there. It says methionine initiation codon, TAC, TAC. Anytime you see TAC, that's where the gene begins, right? Anything after TAC are all the amino acids, that are involved that are making that 1 protein for the body, okay? We also have to have a place to stop. The copying machinery is going to copy the DNA to send to the ribosome. Have to have a place to stop. So we call those termination codons because there are several of them. In fact, there are 3. If you look down the bottom somewhere, there they are right there. There's our termination codons. There are 3 of those: ATT, ATC, and ACT. It's not essential for you guys to memorize all that sort of stuff, but you should know, there is a place you have to start. Initiation, is a codon. We have to have that, and you have to have some place to stop a termination codon. Believe me, like if you're doing this profession, you'll stare at this for so much. You don't have to memorize it. It'll just come in by osmosis by time. Like if I'm looking for something like serine, he's going to say AGC. Yes, of course. There it is. See, I know all different ways to get serine and all this stuff because you just stare at it long enough. So don't worry about having to memorize any of this sort of stuff. It will stick in your head and use up useless space because we utilize, charts like this, computer programs, to do that kind of stuff for it, to get the conception of what's going on. Also, what's sort of interesting here is the body only needs to put together 20 amino acids. Pretty much all organisms are the same. I mean, cats, there's a couple of different things which are sort of out there that are sort of taurine. You may have to add that to get a cat. But pretty much the amino acids across species are pretty much the same. But notice this. It says 9 are essential. Now 11 are made within the body. They're not essential. They're made within the body. And 9 are essential. Now that's a variable across species too. Not much, a little variation. Dogs, you need additional amino acid. No, cats. No, dogs need 1 more, and cats need 2 more. So you get 11. They're essential in the diet. But within 1 or 2 amino acids, they're pretty much the same. Pretty much the same essential amino acids, right? And pretty much the same non-essential amino acids, but very different ways in which they acquire the amino acids by diet. And this is the really important part. Okay, now let's focus on this. All right, this is very important talking about what are humans designed to do. Okay? So when you look at herbivores, things like cows and ungulates, and artiodactyls and all the different ungulates with different sorts of toes, they pretty much have the same sort of digestive system. Okay, they're ruminants, multi-chamber stomachs. Lots of bacteria and colonies of organisms that literally take the vegetative matter, break it down, you know, and pull all the individual molecules, including the amino acids, piece by piece, and make them available to transport across the stomach wall into the bloodstream, and then being uptake by the cells themselves to be assembled into other proteins, larger macromolecules, which the animal body needs. All right. Okay? So it can do that by eating large quantities of vegetables. The ruminant system just goes to work and it can assimilate the amino acids which it needs. Now, as you slide, if you become, let's go to a pure carnivore first. Let's go before intermediate. Let's go to like a big cat, like a lion. Now, a lion's different. They do not ruminate. Okay. In fact, in most vegetable stuff, all vegetable stuff, they don't have the necessary ingredients, within their digestive system to extract amino acids from vegetable matter. It doesn't work that way. They just don't eat vegetables. They get nothing out of it. So their digestive system is to actually quickly transport amino acids from live prey, from meat, okay, from live animals. And they get tons this way. Amino acids just flow across the digestive system. And if you eat an animal, you're getting all of the amino acids, right? All the amino acids that you need, all the essential amino acids, you're going to get, boom, right? That's right into your system. Now, the issue is, now suppose that you're a carnivore, and you're saying, well, how do you get vitamins and minerals? Well, it's because they eat all the animal, the entire animal, right? Through biological magnification, like you have all this vitamin C, vitamin D, everything locked up within your body. It's just in your glands and your organs, not your skeletal muscle. So most carnivores, when they eat you, they're going to eat everything except for your entrails, for like all your junk is at, right? The feces and stuff like that. And by doing that, they're going to get all the vitamins and minerals which they need. That's it. Now, ungulates don't have that option. They don't have that option. That's it. They're limited by uptake. Okay. Their uptake is regulated by the digestive system that they have that actually allows the amino acids. And but watch this. What happens if, through time, vegetative matter begins to decrease in the environment? Okay, not only are you not getting enough calories, but you're not getting enough amino acids. Okay, boom, that's going to lead the organism towards the decline, towards extinction, unless you can accumulate mutations to change the digestive pathway to be able to accommodate different sort of food resources. Now, here's the deal. There's not many other different food resources except for vegetables, meat. You've got to make that shift. And this is a big shift. And once you start committing to that shift, there are big compromises within the system, right? Now, we're part of that compromise. Okay. So by and large, we take a lot of vegetative matter in, we can, but because we shifted a great deal of our diet, our dietary bulk into meat, and we can see that in our evolutionary past. Up until just a very recent time, the past few hundred years ago, most of us were hunters and gatherers, and mostly what we were hunting, was the bolus of our food. We ate everything. Like grandma ate her liver, all that stuff, the testicles and brain and all that stuff we would digest and have a full complement of our diet. And things like vegetables were things we just didn't eat very much. And we're collecting nuts and fruits a lot, for additional calories and fallback food resources when we couldn't hunt to sort of get us by. But the thing about it is just what really where all of our calories and our mainstay was coming from, right? And humans are good generalists because we can fall back on those foods for long periods of time, right? Now, here's the thing, is that, for an ungulate, making this transition across is very difficult, okay? Because primarily, you're going to have to do a real quick switch to your digestive system, right, to compensate, like we are, this half and half thing, right? So you're going to lose the ruminative capacity of the stomach. Therefore, the ability to extract amino acids from most of the vegetable material is going to stop, right? So now you got a problem. That essentially the availability of amino acids, right, is only going to be shifted over towards meat in a very big hurry, because you don't have the digestive system to pull the amino acids out of most of the plants. Now, humans still have some ability, like rice and beans and a few of those things that you can still take some amino acids from, but we don't get them out of many plants. So you got to know exactly which plants to sort of put together to get a full complement, to get all the amino acids that you want, And for most of human history, it's not possible. You can't order this thing here in your hunter-gatherer. I need to have that plant now to mix this plant here to pull this together. That's not part of evolutionary history. It never happened that way because it wasn't possible, okay? Vegetarianism is sort of a product of our recent history, being able to figure out minimal things we can extract from plants and sort of get those together from the grocery store. Okay, so I want you guys to be sort of clear about that. Where in herbivores, it's not like that whatsoever. Okay, so through time, we are selected, right, to actually uptake more of our amino acids. Some people say making more of our amino acids. I guess you can put it in those sort of terms. You know, we make more out of what we see in front without an intermediate, it's direct absorption, you know, versus after having a symbiotic sort of ruminant culture. You know, part of our culture is a little ruminant. We do have an intestinal microflora at work because that's why we can get a lot of the vegetable matter is, but we're far more dependent on our own physiology and getting those essential amino acids. And the bolus is moving towards meat. We're moving towards meat with a lot of failover, right? Now, the reason that we do eat vegetables now is only because of our meat choices. I mean, think about it. We're very cultural. We don't eat all the glands. We've decided to just, you know, I want to eat just this part of the rib or the New York, the loins, the tenderloins back here. Well, all you're eating is the skeletal portion of the meat, you know, which is really just the amino acids. You're not getting any really, you know, vitamins or minerals. So now you have to eat vegetables. Now we got to plant vegetables so you can eat vegetables. So you can get the vitamins and minerals that you need, which were normally in meat. So when we talk about, you know, what the FDA requirements are, that's kind of weird because they really don't understand our evolutionary history, right? And they're not taking into account our cultural choices because they don't want to, because they are a political organization. They already know what you like to eat. So based on what your food choices are going to be, how will you put them together to keep you healthy? Not necessarily what you're actually designed to eat, okay? Now, there is the thought here that amino acids can be actually produced and made, you know, in ungulates. And really what we mean by that, and they do, but remember those being produced and made means more a facilitated transfer from the vegetable matter to the physiology system of rumination into the organism itself. So that's actually what we mean by manufactured and made, okay? So that's what we mean by that. I don't want to make me like they're actually constructed within like the cells themselves, because that's not possible. It's in the intermediate part of physiology, which is still part of it. Okay, all right, I'm going to keep going now. All right. So, you know, we talk about DNA and how it changes, the most important part of the class here, because that genetic code has to change somehow. All those things in those letters, you know, they have to change. If they don't change, everything is static, nothing changes at all, nothing evolves. And nothing evolves to change in new conditions, the species goes extinct. So how do we require changes in that? Well, it just remains changing letters, okay? We have to make mistakes when the code is being copied from one generation to the other. All right, so let's look at this. There are two ways in which mutations can happen, two places I could say, okay? All the DNA in your body is done in the body, but the specific type of cells we got to look at. In general, cells that aren't involved in replication, these are called somatic cells, like, you know, the cells of my fingers, cells of my toes, my nose, and these don't go into reproduction. I can't give my nose cells to my kids. They're all my own, but they do replicate through all their life. Those cells, they keep going on and on and on, and then they stop. And then once they stop dividing, you begin to die, and then you're done, right? Okay, like I'm going through right now. It's horrible. But you all are. After you reach, you know, your certain sort of growth age, your cells are slowing down in their reproduction. And then finally, you're going to get older and older and older. You're all at that age now. Now the aging process begins. Congratulations. Welcome to my world. Here's the deal. You've got a couple hundred, you know, rounds of replications. And each time your DNA divides, create new cells for your body, you know, the DNA is going to create 30 or so, maybe to 40 mistakes that weren't there before, genetic mutations that you can pass on to your own body, you know. So therefore, if I've had maybe a few hundred rounds of replication, there are probably 2,000 to 3,000 different changes, you know, cumulative changes in all my cell generations, you know, 2,000 to 3,000 different changes in each one of the cells of my body, type of cell, skin cell, this cell, that weren't there when I was like a teenager. Thousands of different changes that weren't there. Thus, I can say with all honesty, I'm not the same man I used to be 30 years ago, right? Not physiologically and not psychologically, and not because of experience, right? It's simply because of just ribald genetic expression and the changes in it caused by those mutations as those cells have divided, okay? It's sort of interesting when we talk about the onset of physical diseases or even mental illnesses in your life. They can be wrought on by changes, in the cellular architecture because of mutations within the DNA, right? So you will grow throughout your life. People say, I'll never change as long as I live. I laugh at people. I go, you can't help it. You must change because you are biologically inside out. You cannot stop that or will that stop happening, you know? Therefore, I tell people, that they don't like the life they're living right now. Believe me, it's going to change no matter what you do. Just sit tight because you're going to change along with it. All right. Well, here's the deal. Those changes only affect you. We're interested in changes that happen in your sex cells, right? Those within your gonads. For males in the testes, for females in the ovaries. The primary sex cells, they have to divide. And their process of division, which we call meiosis, creates sex cells. And as that DNA divides, it creates 30 to 40 mistakes that weren't there before that you pass on to your progeny. And your gametes go into sexual reproduction, and that becomes your progeny, right? Well, it's a very simple process. As cells divide, they need complementary DNA. Therefore, the DNA must divide and replicate itself. And how it's done is the strands of the DNA, the individual single strands, just split. One strand on one strand on the other side. And each one of the strands builds a complementary strand. And they separate into two different cells. Now, as they're building that complementary strand on each side, that's when the mistakes are made. Right? So we can change, a single letter. We can make a point mutation. Bam, make a single point mutation, probably change a single amino acid. We can insert a letter, right? That's a big change. It could actually change the reading frame of all the amino acids. If you have three things, everything's going on three at a time, and you insert amino acid, it changes the order of three. All of a sudden, that means every amino acid, from the downline of one spot where you insert a letter will change. Okay. Same thing if you insert two letters at that point, it'll what we call a frame shifter reading error. Make everything down line will be a completely different amino acid. Or maybe you can insert three letters. You can insert an entire amino acid. So it won't change all the letters in the protein. It may just insert an additional amino acid, another great big change. Or we can do the reverse of this. Instead of, inserting letters, we can take them away. We can delete, causing a major frame shift mutation. We can delete two letters, causing a major frame shift, or we can maybe delete three and just remove one amino acid at one point, right? This will then shorten the protein sequence with less amino acid, right? So all these different ways in which we can mutate the DNA to give changes in the structure and function of the offspring's proteins as this building the body and regulating and maintaining its body throughout its life, right? It all happens in only one spot, and that's when DNA divides during the process of meiosis. That's what it's called in your gonads. So remember, you take those two strands, strand 1 and 2, move them apart, split them apart. We'll build a complementary strand on number 1, which would be a duplicate of 2. On strand number 2, we'll build a complementary strand of 1, and we'll lace the 2. But as we build that strand, there's going to be changes in those letters. That's a mutation. That's evolution. It only happens in one spot, and that's in meiosis, in the replication or the division of primary sex cells, in the gonads. Males, testes, females in the ovaries. All right. Okay. So, it's a pretty easy thing to sort of kind of think about if you just sort of hold 4 ideas in your head. Okay. All right. So you separate the strands, right? Then you have enzymes that have to copy, molecular machines, and they're copying the material, adding letters together, and they're going to make mistakes. They're going to make mistakes. Okay. 30 to 40, 3 billion letters, 30 to 40 mistakes. Okay. Well, here's the thing, is that not everywhere in the DNA are mistakes equal. Okay. But where we think they should be randomized and equal, they are not. Okay? So let's look at it this way. Like if I took your hearts, all your hearts out, and tore all your hearts out and put it in front of the desk in front of me, like my ex-girlfriend did to me, ripped my heart out of my chest and just stuck it on the table in front of me, right? And then I gently cut your heads off. I'm going to put them back up. You'll be normal again. You won't even see a difference. I'll put them right next to your hearts. I line them all up in front of me. Where do you guys think you're going to see more difference at? And the hearts are in the heads. In the heads, you're going to see more variation in faces than you are in hearts. Because you can have more variation without calling it a detriment to the organ. If you have so many variations in the heart, you're not going to be a very successful species. Too many mutations in the heart tissue will mean non-viability. But we can have a lot of variation in faces. We see there's 2 ways in which that's accomplished, right? So, in many areas of the genome, we can do a lot of proofreading and repair. And that's where we think regulatory genes, like things like sonic hedgehog, hedgehog, what you saw, in your inner fish, we also call these things HOX genes. That's HOX there. And it's also an acronym for what we call homeobox, right? And all these technical jargon terms for just means it's a regulatory gene. We do think that regulatory genes can sort of direct where to put more proofreading and repair in it, which part of the genome, right? That we want high viability. Don't make too many mistakes like the heart, lungs, vital tissue, right? And where we can have more high variable regions by directing proofreading repair systems. Like in the heart tissue, nope, make sure that copy is pretty close to what we're talking about in the parental copy. Look at it back and forth, proofread it, and repair it if it's wrong, okay? That way we can correct mistakes, preventing the risk of a maladaptation, okay? Or a negative consequence through a negative mutation. Other ways which we think are happening are that regulatory genes can produce algorithms which can direct more randomized mutations in some areas without proofreading. Like, okay, over there, put more random stuff, more than random stuff over here. So we think there are algorithms which seem to control this by the looks, by the way we can see it presenting itself in the organism. All of this stuff is under highly, a big investigation right now. We're right at the cutting edge of where all this stuff is happening, which we're going to be looking at in the next section, which is the regulation of DNA. We'll be looking at that next. Okay, all right. So that's what I have for today. Now we'll get into the regulation of the DNA system, which is far more interesting, I think, and we'll really get to the prime point. What's the prime mover of the system? How is it that change actually occurs in fine, to create this glorious spectrum of all the life that we see around us? As Darwin said, infinite forms, most beautiful and most wonderful, have been and are constantly being evolved. That's the last that paragraph of his entire book, The Origin of the Species. I hope you guys have a chance to read that sometime. All right, so I'm going to stop the share right now, and we'll stop our recording.